Hello everyone, I'm Tony Torres, and I would like to introduce you to the world of silent exchanges, how viral gene swaps shape microbial diversity. Imagine a bustling city where the residents not only communicate, but also occasionally share tools, ideas, or even entire blueprints for survival. This is the microbial world, where silent exchanges of genetic material occur continuously, often mediated by viruses. These exchanges are profoundly shaping microbial ecosystems, especially within our own bodies. Today we'll uncover the mechanisms behind these exchanges, their role in microbial diversity, and their implications for public health. The marvel of horizontal gene transfer in the microbiome. At the core of this phenomenon is horizontal gene transfer, or HGT. Unlike traditional genetic inheritance, where traits are passed down from parent to offspring, HGT allows bacteria to acquire new traits from their neighbors or environment in real time. It's an evolutionary shortcut, enabling microbes to adapt rapidly to challenges. In the human gut microbiome, a dense and dynamic microbial community, HGT plays a vital role. Our gut is home to trillions of microbes, and their genetic exchanges help them respond to changes in diet, immune system pressures, and even external stressors like medications. These silent exchanges create a microbial ecosystem that's not only diverse but also resilient. To put this in perspective, think about how quickly bacteria can develop resistance to antibiotics. Much of this speed comes from HGT, which allows bacteria to borrow resistance genes from others, even if they belong to completely different species. It's a remarkable but double-edged sword. The mechanisms behind the magic. So, how exactly do microbes share their genetic material? There are three primary mechanisms. 1. Conjugation. This is essentially bacterial teamwork. Through a specialized structure called a pilus, bacteria physically connect to each other and transfer DNA. Think of it as one classmate handing another a cheat sheet during a test with all the correct answer, guaranteeing the cheating student a quick and precise A. 2. Transformation. This is the scavenger's strategy. Some bacteria can pick up free-floating DNA fragments from their environment and incorporate them into their own genome. It's as if they're finding discarded treasures on the street and repurposing them to their advantage. 3. Transduction. Perhaps the most fascinating of all, this process involves bacteriophages, viruses that infect bacteria. During their infection cycle, these viruses can accidentally package bacterial DNA and transfer it to another bacterium. It's like UPS, FedEx, or Amazon delivering a package, but mixing up the contents. Each of these mechanisms highlights the ingenuity of microbes in surviving and thriving, often in ways we're just beginning to understand. Effects on microbial diversity and function. Why does this matter? The consequences of HGT are profound and multifaceted, especially in terms of microbial diversity and function. Firstly, HGT fosters ecological adaptation. In the human gut, microbes constantly face challenges, such as changes in diet or the immune system. By getting genes that help them metabolize new nutrients or evade immune responses, these microbes can adapt and keep their place in the ecosystem. Secondly, HGT enhances microbial functions. For instance, it can introduce entirely new metabolic pathways, enabling bacteria to break down complex compounds or interact more effectively with the host's immune system. This is why a healthy microbiome can support digestion, immunity, and overall health. Lastly, HGT ensures functional redundancy. By spreading essential genes across different microbial species, it creates a safety net. If one species is wiped out by antibiotics or illness, others can step in to perform the same functions, maintaining the stability of the microbiome. To put it simply, HGT is like nature's version of a recipe that anyone can view, modify, and share with open ingredients and steps, allowing others to improve and build on it, unlike a secret recipe controlled by the chef. Antibiotic resistance, a growing threat. But HGT isn't always beneficial. One of its darker aspects is its role in spreading antibiotic resistance. Bacteria can acquire resistance genes through conjugation, transformation, or transduction, making them impervious to drugs that once controlled them. For example, bacteriophages can transfer resistance genes from one bacterial population to another, even across different environments. In agriculture, the overuse of antibiotics has created reservoirs of resistance genes that can find their way into human pathogens through HGT. This has turned some infections into treatment-resistant threats, complicating healthcare worldwide. The environmental impact of antibiotic use compounds the problem. Antibiotics in wastewater, agriculture, and industrial processes create selective pressures, encouraging bacteria to exchange resistance genes. What was once a niche problem has now become a global public health challenge. Balancing the benefits of microbial diversity with the risks of resistance requires targeted strategies. Reducing unnecessary antibiotic use and monitoring microbial ecosystems are critical steps in this direction. The tools and challenges of detecting HGT events is no small feat. Traditional microbiological methods often miss these subtle exchanges, which occur on microscopic and molecular levels. However, advances in bioinformatics are providing powerful tools to identify and track HGT. By analyzing microbial genomes and mapping genetic similarities, researchers can pinpoint when and where HGT has occurred. Despite these advances, challenges remain. For instance, understanding how environmental factors such as temperature, diet, or antibiotic exposure influence HGT is still a developing field. Additionally, many HGT events happen without obvious signs, making them difficult to detect and study. Future directions, opportunities for breakthroughs. Looking ahead, there's immense potential for advancing our understanding of HGT. By focusing on the mechanisms of conjugation, the role of bacteriophages, and the environmental triggers of HGT, we can unlock new strategies to manage microbial ecosystems. For example, can we use bacteriophages to target harmful bacteria while preserving beneficial ones? Can we design antibiotics that limit HGT without harming the microbiome? These are exciting possibilities that require interdisciplinary collaboration. In conclusion, the silent exchanges of genes within the microbial world are a testament to nature's ingenuity. They drive diversity, enable adaptation, and create resilience within microbial ecosystems. At the same time, they pose challenges, particularly in the context of antibiotic resistance and public health. By understanding and managing these exchanges, we can harness their potential for good while mitigating their risks. This is a field where microbiology, medicine, and technology intersect, offering opportunities to not only advance science, but also improve human health. Thank you for your attention. I hope this journey into the microbial world has sparked your curiosity and appreciation for these unseen exchanges that shape life as we know it.